Hello, 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 everybody. How are you? Welcome back to my channel Bioscience Hub, my dear friends. Yeah, I know there was a big gap. Okay, there was a big gap between the first video and the second video, but I'm sorry there were some technical issues. But now I will try to be consistent with my videos. Okay, so my dear friends, without wasting much time, we are starting with the second lecture of Animal Kingdom. Okay, we are starting with the second lecture of Animal Kingdom, but before starting we need to revise. Okay, because revision gives acceleration to our mind to recall the previously learned things. Okay, therefore we are going to start with revision. Okay, so what we have learned in the previous class, we learned a very few things in the previous class. Okay, what were the things that there are about over a million species present on the earth. Over a million species are present. Over a million species are present on the earth. On earth. Now, because there are more than million species, studying them combinedly won't help. It would be a hectic work, or it would be a headache for the scientists to study them combined. So therefore what they did, they classified these million species. Okay, how did they classify these species? They classified these species as amphibians, okay, reptiles, fishes, and mammals. Just before moving forward, let me check whether you can see the board or not. Means you can see the board, but can you see the written things on the board clearly? Yeah, they are clear. I'm sorry because okay, I'm sorry if there is some problem because I have health issues right now. Okay, so please bear with me. Okay, so these million species are classified on the basis of on are classified in groups of amphibians, reptiles, fishes, and mammals. Okay, this we studied. After this, we studied levels of organization. What did we study? We studied levels of organization. Okay, so what were the levels of organization? First level was cellular. After this, we studied levels of organization. Let me put it in a box. Okay, in cellular level of organization, I see I'm not going to explain these levels of organization again because reading the name of organization, you can figure out cellular means made up of only consists cell. The organism consists only cell. In cellular, we studied sponges. Okay, from the phylum Porifera. Okay, now you don't have to what we say. Now you don't have to focus on the phylums because I'm going to conduct a lecture special. I'm going to conduct a lecture on each and every phylum. In the third or fourth lecture, in the upcoming lectures, we are going to study the phylum. So don't worry about the phylums. I'm just telling you. After cellular, we studied what we studied. We studied tissue level. Okay, we studied tissue level. In tissue level, we saw jellyfish and hydra. We saw the examples of, just a minute, I'll write it over here. Jellyfish and hydra. From the phylum, you don't have to write anything. You just have to close your books and you have to recall the things with me. Okay, jellyfish and hydra were the examples which we studied in the phylum. Sorry, in the tissue level organization. Which phylum do they belong? They belong to the phylum Nidaria. Okay, I'll write it over here. Nidaria, not Nidaria, Nidaria. Okay. After this, we studied organ level of organization, cellular, tissue. After this, organ level of organization. What examples did we see in organ level of organization? In organ level of organization, we saw the example of planaria and platyhelminths. Just a minute. Yeah. In organ level, in organ level of organization, we saw the examples of platyhelminths and planaria. Platyhelminths and planaria. Okay. And after this, the final level of organization which we studied was organ system level of organization. That I am writing over here. Okay. So, organ system level of organization. LOO means level of organization. This much you know. Okay. In this we studied some roundworms. In this there was example of roundworms. Okay. And vertebrates. Okay, in this we studied roundworms and we saw the examples of vertebrates. Okay. After this, we saw, what did we see? That in platyhelminths, okay, Jashan, the digestive system, we saw the digestive system of the platyhelminths. Okay, so the digestive system of platyhelminths is a very simple type. See, there is only one opening. There is only one opening which is responsible for the intake of food and for excretion. Okay, means only one opening eats the food, and with the same opening, the food, the excretory material, the excreted material is thrown out. Okay, so this is not a complete digestive system. Okay, the complete digestive system means an individual and a specific opening for intake of food and a specific opening for excreting the waste material, which is mouth and anus. Okay, means with mouth we eat and with anus we excrete out the waste material. Understood? After this, just a minute, let me erase the board. Just give me a minute. Okay, we saw, after this we saw the circulatory system. We saw the circulatory system. Okay, the circulatory system is divided into two parts. The circulatory system is divided into two parts: open circulatory system and closed circulatory system. Okay, open circulatory system, the fluid is called hemolymph. Fluid, hemolymph. Okay, so now tell me, why is it called hemolymph? Because this fluid lacks the main components of blood. Even if it is composed of blood, it lacks the main components, which are RBCs, okay, and platelets, which are RBCs and platelets. Okay, after this, what we studied? After this, we studied that the heart. Okay, the heart. Sorry, the hemolymph gets pumped from the heart, gets pumped, and the tissues and the tissues, organs or cells directly bath into it. Okay, means there is no special construction, that what we say, like we have, there is no special construction as of us, that there are arteries, veins, the these sorry, I'm sorry, I'm really sorry. So these tissues, organs or cells gets directly dipped into it. Okay, understood? Arteries, veins and capillaries are absent. Arteries, I'll write it here. Arteries, veins and capillaries are absent. Okay, arteries, veins and capillaries are absent. Just a minute. Yeah. But what is in closed circulatory system? The closed circulatory system consists of arteries, veins and capillaries. Okay, and what we say is, sorry, just a minute. Yeah, just a minute. Let me write the examples. I didn't write the examples of open circulatory system. Yeah, no, I'm sorry. After this, what did we study in the closed circulatory system? In closed circulatory system, we studied that there is a network of capillaries, network of capillaries, veins, just a minute. I'm sorry. Network of capillaries, veins and arteries are present. And in what organisms do we see closed circulatory system? We see closed circulatory system in annelids. Correct? In annelids? Yeah. We see closed circulatory system in annelids. Example is annelids, all the vertebrates, and what do we say? All the vertebrates, fishes and mammals. Okay, all the vertebrates, fishes, and all the vertebrates and fishes and mammals, and fishes, comma mammals. and fishes, comma mammals. Okay, so this was the thing. This was the total summary of last lecture which we did in 15 minutes. Okay, so now we are going to move on to the next. I'm not singing the song. If you're done with it, next. Move on to the next. Okay, but now we are going to start with the next portion. Okay, just a minute. Just give me a minute. So now, moving on to the next, we are starting with the circular. I'm sorry, with the symmetry. Okay, so just concentrate over here. What do you mean by symmetry? Symmetry, in short, means dividing something in two equal halves. Okay, so now we are going to study the same. Okay, what I have written that? Yeah. So the scientists not only the scientists not only use the what we say levels of organizations criteria, but they also use some more criterias like what we say is symmetry, uh, germ layers, and coeloms means body cavity. So symmetry is very simple because you have learned symmetry not of animals but of other things in lower classes. So symmetry is divided into bilateral three types: bilateral, radial, and asymmetry. Okay, bilateral, radial, and asymmetry. So now, what is this asymmetry? First of all, let's start with this. What is this asymmetry? This asymmetry, when you pass a plane, when you just a minute. When you pass a plane and the organism is not divided, organisms cannot be divided, then it is called asymmetry. Okay, what are the examples of asymmetry? Sponges, correct? Yeah. Now just let me tell you, sorry, cannot be divided into two equal halves, cannot be divided into two equal halves. Okay, it is called asymmetry. Example is sponges. Okay, example is sponges. After this, did you understand? Means, see, this is a sponge. Means this is not an organism, but let's check it. When you put a plane in it, it cannot, if it does not get divided, it is asymmetry. Understood? Okay. Now what is radial symmetry? Radial symmetry through center when we pass, okay, through the center from top to bottom when we pass a plane and get to and get two equal parts, it is called radial symmetry. What could be the example of radial symmetry? The example of radial symmetry are some sponges, cnidarians. Okay, just a minute. Example. Just give me a minute. Some sponges. I'm not saying all other or your NCRT not at all says that all the sponges. Okay. Some sponges, echinoderms. Where should I write? I write it over here. Echinoderms. Okay, echinoderms, coelentrates, ctenophores. Okay, sorry, echinoderms, cnidarians, and ctenophores. Echinoderms, cnidarians or cnidaria, and ctenophore. So these are the examples of what we say. Um, these are the examples of radial symmetry. How much time do we have? Oh, we can get time to for the question practice also then. Okay. Bilateral symmetry. Now moving on to the bilateral symmetry. What does the bilateral symmetry says? Okay. There is a central axis and the organs are paired. Okay, and the organs are paired. Understand me what I'm saying? Okay. And the organs are paired on both the sides. On both the sides. Okay. Can be divided into two halves. Can be divided into two equal halves. Okay, this is called bilateral symmetry. What are the examples? The examples of all the examples are many invertebrates are the example. Many, I'm not saying all, or your NCRT does not at all says all the invertebrates, but it says all the vertebrates. Many invertebrates, and all the vertebrates. Just a minute. Let me divide it. You won't understand. Can both the sides can be divided into two halves, into equal two halves. Okay, this is the first part. Okay, understand? Understood? Are you getting or not what I'm teaching or what I'm trying to explain you? After this, understood, completed. Symmetry chapter, symmetry portion is completed. Now, let's move on to the next part. Okay. Now, what are we going to study the next part? The next part which we are going to study is we are going to study, okay, the germ layers. Okay, I'm sorry, I couldn't recall it for few minutes, few seconds, sorry. What are we studying now? We are studying germ layers. Okay, so my dear friends, the germ layer is again of two types. Okay, so now the classification criteria of organisms used by scientists was a new one which is called germ layers. So the germ layers are divided into two parts: diploblastic, diploblast or triploblast, and triploblast, not or, and triploblast or triploblastic. Okay, diploblastic and triploblastic. So is the mic on? Yeah. What do you understand by the word diploblast? Means there are only two germ layers present. Only, I'll just write it a big, write it a little big. Only two germ layers are present. Okay. What is present? Only two germ layers. Try to understand. Only two germ layers are present, and those germ layers are ectoderm and endoderm. And when are these germ layers formed? These germ layers, just a minute. These germ layers are formed at the embryonic level itself. At the embryonic level itself. Okay, at the embryonic level only. Got it? So now see here, how are these germ layers? Let me tell you. This is one, and this is one. So this layer is called ectoderm, and this layer is called endoderm. Okay, and in what organisms do we see diploblastic germ layer? Okay, means diploblast organ. We see diploblastic germ layer in coelentrates. Example I'm writing here. Example is coelentrate. Okay. Whereas now in triploblast, everything is same, but there is a jelly-like structure. Okay, there is a jelly-like structure. Just a minute. I'll. Okay, so there is an jelly-like undifferentiated layer, jelly-like structure or undifferentiated layer present in triploblastic. Okay, just give me a. Okay, so now we trying to understand the triploelastic germ layer. Okay, now triplo, obviously three germ layers are present. Three germ layers. Okay, ectoderm, endoderm. Sorry. Okay, ectoderm, endoderm, and one more jelly-like structure, and a jelly-like structure. I'm sorry. Okay, and a jelly-like structure called mesoglia. Okay, jelly-like structure or called jelly-like structure. Okay, and a jelly-like structure or a. I'm sorry, I was a bit wrong. Or a gel or a mesoglial layer called mesoderm. Ectoderm, endoderm, mesoderm. And my dear friends, this mesoderm is connected. It is an undifferentiated, means it is connected to this. Okay, this is called mesoglia. This is called mesoderm. Okay, I hope you can see. Just a minute, I'll just write it with a red pen. It is called mesoderm. Okay, so it is where it is present. Let me tell you. Between ectoderm and endoderm, I suppose this is the position or this is the position. Okay, so like this, there is a mesoderm, ectoderm, endoderm, and mesoderm. Understanding? Just give me a minute. I'm sorry, I went wrong. Okay, see here. Let me confirm. You all are pausing the video and taking down the notes, correct? Even if you're not, you will be getting the lecture notes now. Okay, as soon as the link verification is completed, you will be getting it. Okay, so see here, where is this mesoderm layer present? This mesoderm layer is present here. See, this is ectoderm. Okay, here it is endoderm, and in between the layer, and in between there is mesoderm present. Okay, so this is ectoderm. Okay, this is, the blue pen isn't working. Just a minute. So see, this is ectoderm, this is ecto, this is endo. Okay, this is endo, and in between this is mesoderm. Okay, and same, this triploblastic triploblastic organization is also formed during the embryonic level only. Okay, with this. Okay, so now just let me give you the examples. I gave you the example of sealant traits. Okay, sealant traits. Okay, and the example is just a minute. Example is platyhelminths to chordates. I'll write this example over here. Example, platyhelminths to chordates. Okay, triploblastic. The example of triploblastic is written over here. Okay, with this we completed with diploblastic and triploblastic also. Now moving on to the next, we will be studying coelom. Okay, now what are we going to study? We are going to study coelom. What is the coelom? Okay, coelom, in other words, in a simple word, you can say it is a body cavity. Coelom is, in a simple word, body cavity. Okay, lined by mesoderm. Now this coelom is divided into three parts. Coelom. Okay. Acoelomates. Acoelom. Okay. Acoelomate. Pseudocoelomates. And coelomates. And coeloms. Now in this acoelomate, A means absent. A means absent, means mesodermal line. Okay, means in this the mesoderm lining is absent. Okay, in this the mesoderm body cavity is okay. In pseudocoeloms, the body cavity is not completely lined. Body cavity isn't completely lined by mesoderm. Okay, means it's partially. Isn't completely, means partially. This much you can understand that if I'm writing isn't completely, then it then it what we said directly, it means that it is present partially. Or else I'll write is a body cavity is partially is okay. So it is partially lined by mesoderm, and in coelomates, body cavity is present. Okay, body cavity or mesodermal lining is present. Okay, body cavity mesodermal lining is present. Okay, so now what are the examples? The examples are example of this is planaria. Just give me a minute, please. I'm a bit headache. Okay, the example of acoelomate. The example of acoelomate is planaria, means flatworm. Now I'm writing over here. Planaria, flatworm. Okay, Liver fluke, not only flatworm, liver fluke is also there. All these examples we are going to study in the upcoming lectures of phylums. Okay, so don't worry. Hmm. After this, tapeworm also. It is called tinea or tapeworm. Means acelomate means mesoderm lining is absent in platyhelminths. Phylum. I won't write it here. I will write it over here. Platyhelminths. Okay, platyhelminths. After this, coming on to pseudocoelomate, means it is partially lined. It's not completely. It's not in this the lining mesodermal lining is not completely present also and not completely absent also. It is partial. Okay, so what are the examples? The examples are Ascaris wucheria. Okay, Ascaris and wucheria. The examples are Ascaris, which is called roundworm, and wucheria. Okay, and what? And wucheria, it is called filarial worm. Filarial worm. Don't worry about anything. We are going to study it. Okay, don't worry. We are going to study it. After this comes the final. What we say is coelomates. Okay, in this, the examples are earthworms, cockroach, and leech. Okay, earthworm, earthworm is called the farmer's friend. Cockroach, every time when you switch off the lights of the kitchen and in the midnight when you see, okay, when you switch off the lights of the kitchen in the night at night, and in case if you get up searching for the food at night, if you fought with your parents, if you have a fight, if you have a wording, if you have a word fight with your parents, then you sleep most of the time. After fighting with your parents, with the parents, we sleep hungry. Okay, we say we will not eat. Okay, but at night we feel hungry, and uh not a very small amount of hunger we feel a very large amount of hunger. So when we wake up after feeling hungry, when we wake up at night for searching for the food, and when we turn on the lights of the kitchen, we see the cockroaches running from here and there, that they are racing with each other, and as soon as the light turns on, they start running more quick. Okay, so that's leech is called blood sucking animal. Leech is called blood sucking animals. Okay, uh, eh, in ayurvedic what we say, in ayurvedic treatments, leech is used. In ayurvedic treatments, leech is used. It's wherever you get a wound, if you get it over on the hand or on the thigh or any of the body part, and you have, and you need to suck out the uh, what we say, the impure blood. In ayurvedic hospitals or in ayurvedic therapies, in ayurvedic treatments, leech is used. Okay, leech is placed, the leech starts sucking. Okay, it what we say, it releases it releases a type of chemical where, okay, and it starts sucking the blood. Okay, so these are the examples. Understood? If you want, you can pause the video and write this down. Okay, now moving on to the last but not the least and the very easiest part, segmentation. Okay, sorry, segmentation and notochord. Okay, so notochord and segmentation are a very simple topics. So these are the last two topics of today's lecture, segmentation and notochord. Okay, so segmentation is divided into two parts. Segmentation is a meristematic. Okay, is it written meristematic? Just a minute. Okay, sorry. Okay, segmentation. This segmentation is divided again into two types: external and internal. Okay, so in external segmentation, okay, rings are visible. Okay, in external, we can differentiate. We can differentiate with the eyes. We can see. We can see the external segmentation with our eyes. Okay, whereas in internal segmentation we can't see. Okay, internal we can't see, and internal segmentation is done by septa. Oh. Internal segmentation is the body is divided from inside, from inside by a septa, by a septa called internal wall. What is this septa? This septa is an internal wall. Okay, this septa is an internal wall. Done, completed. Simple examples: cockroach. Example of external: cockroach. There are segments, no? First segment is head, second is thorax, and third is abdomen. So these are called these segments. Okay, just a minute. Okay, this is called segmentation. And internal segmentation, simple, it is seen in, okay, the example of internal segmentation is earthworm. Example is earthworm. And this earthworm, okay, shows a meristematic segmentation. Okay, this earthworm shows a, sorry, sorry, not meristematic, metameric segment. Okay, earthworm shows metameric segmentation. Metameric segmentation. Earthworm shows metameric segmentation, or we can say metamerism, or metamerism. Okay, done. Completed with segmentation. Okay, completed with the segmentation topic. Now, mm, the last topic of today's lecture. Just give me a minute. The last topic of today's lecture is notochord. Okay, now we are going to study notochord. So the last topic, notochord. Okay, this notochord, simple understanding. You don't have to make it complicated. I'm also not going to make it complicated. In triploblastic organisms, okay, in triploblastic organisms, we saw three layers: ectoderm, endoderm, mesoderm. So this notochord is a rod-like structure. A rod-like structure. Just a minute. A rod-like structure, mesodermally derived. Okay, a rod-like structure mesodermally, which is mesodermally derived. Okay, which is mesodermally derived. Okay, where is this notochord? This notochord is present dorsally. It is present dorsally. It is present dorsally. Okay, not in all the animals. In some animals, it is present dorsally in some animals. Okay, and the organisms who possess notochord, organisms who possess notochord, I'll write it over here. Organisms who possess notochord are called chordates. Okay, are called chordates. And organisms who don't have this structure, the notochord absent in organisms are called non-chordates. Okay, these are called non-chordates. What is this? Okay, example is given of non-chordates: platyhelminths, to echinoderms. Okay, platyhelminths to echinoderms. With this, we have completed with our lecture number 2 all the 5 topics. Okay, and revision also of the previous lecture. So thank you everybody for watching this lecture. I hope every one of you understood this, and if there is some issue, please let me know in the comment section, and please like, share, subscribe the video, because your support means a lot to me. Okay, when you all view the channel, okay, when you all view the videos of mine, I get motivation. Okay, and with the same motivation, I'm ready for the new lectures. Okay, thank you everybody. Thank you, thank you, thank you.